Hello, I'm Phillip Sharp, a professor at MIT in the Department of Biology. I have been at MIT now for almost 40 years, working with students and teaching and doing research on how genes are regulated, turned on and turned off, both in cancer and diseases of that nature, as well as in normal conditions. Particularly interested in how genes are turned on and turned off in human cells related to cancer. I came from Kentucky. I grew up on a small farm in Kentucky, lived on the farm until I completed my high school education and then went to a small college, liberal arts college in the mountains of Kentucky, and then went to the University of Illinois to get my PhD in chemistry and mathematics. I particularly loved, as a young student, math and chemistry. I found it a very enjoyable way to learn about the world and relate to the world. But it wasn't until I was in graduate school, getting my doctorate in chemistry, that I really became engaged with understanding the molecular and cellular processes that underlie life systems, how cells grow, how they change in their character, how they use sugar and metabolism to support their growth. That led me on a journey to Caltech, where I was at 2 years doing research, and then to Cold Spring Harbor Laboratory in New York, where I worked with Jim Watson, the Watson and Crick discoverer of the structure of DNA. And I was at Cold Spring Harbor when I began to work on the question of how viral genes actually cause cancer. And I decided I needed to know more about how genes were expressed in cells. You've heard from Eric that, genes are made of DNA, and DNA is copied into RNA, which then is used to be decoded to make protein. And that system had been well worked out and understood in simple organisms, single-cell organisms like bacteria, but really had not been well established in more complex cells such as human cells. And that led me, after I came to MIT and joined the Center for Cancer Research, to turn to the question of what was the structure of genes and how they were expressed in human cells. And I made a very striking discovery, which I didn't anticipate. I thought there would be an interesting answer there, but I didn't anticipate how exciting and different the answer was. And what we were able to discover was that in human cells and many other cells of higher order organisms, the genes come in discontinuous segments. Instead of having all the information in one consecutive series, your genes are structured to be split. They're split with a set of sequences called introns, and the intronic sequences are actually removed at the stage of RNA. So let me illustrate that. I've drawn on the board here what your genes look like. This is a segment of DNA. And here would be a set of sequences that are coding for one of the functional proteins in your cells. And those sequences run continuously here, and then here, and then here. We call those exons. And these sequences in the middle are nonsense, and they have to be removed at the level of RNA to make the functional protein from this gene. So the genetic material in your DNA is edited by RNA splicing to assemble the information that is used to make the proteins that make your muscles and your blood and your other parts of your body. Now that's really amazing, because this whole splicing process, which assembles this information, you can have mutations in the DNA that inactivate that. And in fact, many of the mutations that cause human disease, I'm sure Eric has mentioned this, are those types of mutations in the gene. But it's even more exciting than that. As Eric, I'm sure, told you, there are 23,000 genes encoded in the human genome. But when we look at each of those genes, what we find is that in almost all cases, those genes can be interpreted at the level of RNA in different ways. We call this alternative splicing. Let me explain what that is. In many cells, sequences like this would be spliced together in a regular pattern, making one protein. But in other cells, sequences would be spliced like this, skipping this set of sequences, and that would make a different protein. So, and we call this alternative splicing. And though you have only about 23,000 genes, we know that those 23,000 genes can make a much larger set of proteins that are really important for how the human body carries out its functions. So alternative splicing has increased our understanding of the complexity of the genome and how to interpret that complexity in terms of function. Now, we know in cancer, changes in gene expression are integral to the cancer process. When we take the cells from a human cancer and sequence the genome of those cells, we find hundreds of different mutations in genes. Some of those mutations create genes that drive cancer, and some of those mutations inactivate genes that block cancer. And those mutations controlling genes is integral to how cancer occurs. And when we identify those types of genes, then we can develop drugs to them and impact on the survivability of cancer, potentially moving towards a cure for cancer. We can't cure most cancers now. We can benefit patients with these types of drugs, and we will get better and better at doing it. But to actually develop those drugs, we have to be able to understand how genes are expressed and how genes are regulated, turned up and turned down. MIT is a very special place. We get remarkable students to work with us here at MIT, to work with us in classrooms, understanding and creating material, to work with us in research laboratories as undergrads and as graduates who are getting their PhD. And we have a very stimulating environment to share that knowledge with, because most of the professors at MIT interact with each other and their students interact with each other. So it's a community of learning. It's a community of trying to cure cancer. It's a community trying to understand the basis of neurological problems such as schizophrenia and manic depression. It's really an absolutely fascinating place to create new knowledge. And when we're setting and dealing with a research topic, every student, every professor is sitting there trying to understand how to solve this problem. And anyone who can make a contribution to it is welcome. Welcome from students who are trained here in this country, and welcome to students who are trained around the world in other places.